Muscle contraction. Muscle contraction is actually a much more complex process than you may have first imagined. There are a number of cascading events which result in the movement of skeletal muscle. Contraction occurs as a response to a stimulus, whether it be voluntary, such as during exercise, or involuntary in the form of reflex action. Muscles are comprised of large bundles of long, thin cells known as muscle fibers. Surrounding the fibers is a continuous plasma membrane called the sarcolemma. Within each of the fibrous cells are many myofibrils running in parallel columns. These are made up of two types of proteins with differing properties. One of these proteins is myosin. This is a thick filament with many globular heads resembling golf clubs and can be viewed under an electron microscope as darker areas. The second type of protein is actin. This is a thin filament responsible for the light area seen under an electron microscope. It has a binding site for the myosin head which, when bound, creates an actin-myosin complex or a cross bridge. Together, the actin and myosin protein filaments create single units called sarcomeres. These make up the myofibril. Processes within these units allow for muscle contraction via the sliding filament model. This is when the actin and myosin filaments slide over each other with the aid of a third protein, troponin. Troponin binds to tropomyosin, a protein which blocks the binding sites on the actin filament. The binding of troponin secures the blocking mechanism. Muscle contraction cannot occur unless the tropomyosin is removed from the binding site. The process begins with an action potential which triggers an influx of calcium ions into the neuron, which stimulates the muscle cell, causing depolarization along the length of the fibers. Calcium ions then move into the sarcoplasm, the specific name for the cytoplasm of a muscle cell. These calcium ions bind to the tropomyosin molecules, which triggers a change in shape, causing the tropomyosin to move out of the binding site. It is now exposed for the myosin head to form a cross bridge with the actin filament. Once bound, the movement of the myosin, which pulls the actin filament along, is powered by the hydrolysis of an ATP molecule. The ATP molecule binds to the myosin and is then broken down into ADP plus phosphate, which provides the energy required for movement. A second ATP molecule binds, causing the detachment of the myosin head from the filament, and is also hydrolyzed, re-energizing the head. The cycle is able to begin again further along the filament at another myosin head binding site. This repeating motion shortens the sarcomere and is known overall as contraction. As long as the stimulus continues and calcium ions influx, the process will repeat. So how do cells know when to stop this cycle? Termination occurs when stimulation stops and calcium ions move out of the sarcoplasm via active transport. The troponin now returns to its original form, pulling the tropomyosin molecules back into the binding site and hence blocking it. The myosin can no longer form a cross bridge and contraction is inhibited.